My name is Stefan Kurtenbach, and I'm a research assistant professor at the Bascom-Palmer Eye Institute at the University of Miami in Florida. My main research focus is a cancer in the eye called uveal melanoma. Most people have heard about skin melanoma, which can be caused by too much sunlight exposure, for instance, but far less people have heard about other tumors starting off with a melanocyte, for instance, uveal melanoma itself. Despite sharing the same cell of origin, uveal melanomas are a very different tumors from the more common skin melanomas. This is true in terms of the genetics, in terms of the mutations we see, in terms of how these tumors behave and how they can be treated. There are some treatment options available now for skin melanoma which work pretty well. However, this is not the case for uveal melanoma, unfortunately. Almost 40% of the patients will develop metastases to the liver, which are notoriously hard to treat, and there's really no good treatment option out there to date. And most of those patients will eventually die from this disease. This is what fascinates me about this cancer, that despite decades of research, we're still lacking good tools to treat this cancer and prolong the lives of patients. The last couple of years were very successful for uveal melanoma research. We and other laboratories have characterized those tumors in terms of their genetics, their mutations, their epigenetics, gene expressions, and many other things to understand the things that happen to transform a normal melanocyte in the eye into a tumor cell. Luckily, uveal melanoma is a very simple tumor regarding its genetics, meaning we see a very low mutation burden, and we see mutations in only a very few key genes that we are very aware about. For instance, mutations in a gene called BAP1 are correlating with high metastatic risk and are present in most of the tumors that will form metastasis and patient death, unfortunately. What is true for BAP1 is also true for the other mutations we find, is we understand they're there and we understand some of the roles they're playing, but there's still a huge gap in knowledge in understanding why those mutations are important in forming metastasis for uveal melanoma. In my mind, the lack of treatment options for uveal melanoma is caused directly by our lack in knowledge on what's happening in those tumors themselves. So this is the research focus of our laboratory. We're trying to understand what are these mutations doing for the tumors, and can we exploit them somehow by using drugs. A while back, we have conducted a study to determine whether we can subdivide the risk groups in uveal melanoma further, a little more granular. We know that if a tumor harbors a mutation in BAP1, that is high metastatic risk, and if the BAP1 mutation is not present, that is low metastatic risk. However, we knew that even in the low metastatic risk group, there's a subpopulation of patients that will eventually metastasize, even though later on. And it turns out that this subgroup can be distinguished from the first one by the expression of a single gene, and the single gene is called PRAME. PRAME is actually such a powerful biomarker of metastasis that it's now incorporated as an independent biomarker in the classical clinical tests that are being used to give patients a more definitive answer on how high metastatic risk their tumor really is. Finding this single gene, PRAME, expressed in high-risk uveal melanoma tumors and metastasis is interesting for several reasons. First, PRAME is not expressed in normal tissue types in the body, except in one, the testis. Meaning, if targeted therapies were to be developed for PRAME, one could expect low side effects compared to normal chemotherapy approaches that are being used right now, which really have a broad effect on the body and various organs and tissue types. Second, it is usually easier to target something that is overactivated or overexpressed than something that is inactivated, like in case of BAP1. Given those findings, we are making understanding the role of PRAME in uveal melanoma metastasis a major focus in our laboratory. We're interested in two major questions. The first one being, how is PRAME becoming aberrantly expressed in tumors? Given that PRAME is not expressed in most human tissue types, clearly there's a very specific regulatory mechanism keeping PRAME in check and keeping PRAME from being expressed and causing problems in the cell. We want to understand this mechanism to see if this can be exploited to reduce PRAME expression in tumors. Second, we want to understand what PRAME does once it becomes expressed in tumors. And what we're finding is that PRAME serves specific functions in maintaining the chromosomal and DNA architecture in tumors. Now, when PRAME is expressed, this initially causes problems for the tumors, and there's an adaptation phase. But once the cells overcome this phase, it's actually a pro-cancer mechanism for the cell to adapt to new stimuli and to develop new, more aggressive subclones. To gain insight into this mechanism, we're using several epigenetic methods as well as animal models, which allow us to inject uveal melanoma tumor cells into the eyes of mice and follow them in the live animal as they disseminate and form metastasis in the liver. What we find that this specialized function in DNA maintenance is exploitable by specific drugs. And by applying those drugs to the mice, we find that the mice suffer from lower metastatic burden and live longer. In summary, we describe a new mechanism by which PRAME fosters the development of metastasis in uveal melanoma, and also show a new treatment angle by which the actions of PRAME might be exploited to prolong the lives of patients suffering from uveal melanoma.